Hello, and welcome to Anatomy and Physiology on Demand. These are being pre-recorded so that you don't have to sit and listen to an hour and 15 minutes at one time. Will break them up into multiple little segments, and so you can watch them at your leisure whenever you want. Where we are picking up today is cardiac and smooth muscle, and basically we're going to be comparing and contrasting how these two different types of muscles are, as opposed to the 2 weeks we spent learning all about skeletal muscle. So starting on, cardiac muscle, as you know, is only found in the heart wall. It is involuntary. We will learn later on it's going to be innervated by part of the nervous system known as autonomic motor. However, nervous stimulation is not required for cardiac muscle to contract. Looking at the heart muscle itself, there are these specialized cells, which are hybrid cells between nervous cells and muscle cells, that form a conduction system. There's a pacemaker area which sets the contract rate, and it then just passes from one cell to another cell to another cell through the conduction system. And because it doesn't require any outside input in order to be stimulated to start this process, we use the term autorhythmicity to describe the heart. However, there is nervous input, and what this nervous input does is can increase your heart rate or decrease your heart rate, or it can increase the force of contraction. Now, these are all concepts you're going to learn a lot more about in the second semester, and you come back and you learn about the cardiovascular system. So just a quick review of the histology. Cardiomyocytes are branched, they have striations. We've only mentioned that they are uninucleated. However, I do want you to realize that a significant number of cells do have two nuclei. They are never having more than two nuclei, but they can be uninucleated or binucleated, as seen in the picture. There are spaces between the cells as a result of the branching, and you can see a little bit of fluffiness between these cells. And what that fluffiness, those little tiny strands, are representing is the fact that there's endomysium that surrounds the individual cells. However, there is no perimysium or epimysium surrounding the heart itself. Looking at intercellular junctions, cardiac muscle has two different types, each having a totally different purpose and each equally important. The first are the desmosomes, which have the normal structure of a desmosome with the linking proteins between the two plaques on adjacent plasma membranes, or sarcolemmas, from which we have cytoskeleton taking the tension to distant areas of the cell. And this is important because if we have a vigorous heart muscle contraction, we don't want those cells to rip apart from each other. The second type of junction are the intercalated discs. And what these are those dark areas that you see on the histology under the light microscope. And what they represent are rows of gap junctions. And what this enables is when we have ion changes, either as a result of depolarization or repolarization, when that occurs in one cell, those ions will almost immediately flow into the adjacent cell to cause the same type of ionic change in that second cell. And this is a feature that really helps enable the entire heart muscle area to contract as one rather than sequentially. Now, because the heart muscle has to work 24/7, 365, it needs to have a continuous source of energy. And it's almost exclusively aerobic. There's a robust blood supply, and if we block that, then we're not going to have good muscle contraction. The cells themselves have a lot of myoglobin, lots of stored glycogen, and they have mitochondria, which are significantly larger than mitochondria seen in other cells. And not only that, about 25% of the cell is filled with mitochondria. If you look at the electron microscope picture at the top, you can see that there are just rows of these huge mitochondria between the adjacent myocardiocytes, which you can see with those sarcomeres. And you can go back to previous pictures that we posted of skeletal muscle and compare the two, and you'll see they don't look like each other at all. And this enables the heart to be highly resistant to fatigue. So when we look at the cell, as you just saw, we had sarcomeres. So we have the same actin and myosin arrangement. We have the same troponin and tropomyosin attached to the actin, and calcium binds to troponin, pulling the tropomyosin off. All of that is exactly the same as in skeletal muscle. However, there are some significant differences, okay? Looking at the picture, you can see the T tubules here, but what you should notice is they're quite a bit wider, and they are not part of a triad process. In other words, the adjacent sarcoplasmic reticulum does not have this huge terminal cistern on both sides of it, forming this triad. Instead, what we have is we have little tiny terminal cisterns of the sarcoplasmic reticulum, which attach on one side, and then on the other side of the T tubule, you'll have other ones. And so these are giving us two things forming this connection. So instead of calling it a triad, it's called a dyad. So if you haven't done this recently, and hopefully you did, since there was a quiz on it today, take this chance to review depolarization and repolarization and the sodium-potassium channels of skeletal muscle so that you can understand the differences, what we're going to be talking about today. So, just like in skeletal muscle, we have sodium, potassium, and calcium channels. However, the calcium channels were not involved at all in skeletal muscle contraction, but they are massively involved in cardiac muscle contraction. And this is because when calcium enters the cell through these calcium channels, it's going to act as a second messenger inside the cell. And as you recall, second messengers take time because something else has to happen. And so this results in that we're not going to have an immediate quick little twitch just like we had in skeletal muscle. It's going to maintain the tension longer. It's going to have more calcium ions come in, and it's going to allow blood to be expelled. Okay. So in order to understand this, there's three little concepts I want to introduce one by one before I put the three of them together in cardiac muscle contraction and relaxation. And the first concept is to introduce a new receptor called the ryanodine receptor. And this is actually not on the sarcolemma, but it's on the sarcoplasmic reticulum. And it's quite a long receptor. The vast majority of it is in the cytosol, and then some of it goes through the phospholipid bilayer of the sarcoplasmic reticulum. And what happens is it recognizes the presence of calcium in the cytosol. And so when the calcium level goes up, then this is going to be activated. And when this receptor is activated, it then opens a gate in the sarcoplasmic reticulum, which allows calcium, which was sequestered in there, to be released. Okay, and so now this releasing calcium is going to cause a rise in calcium concentration in the cytosol even more. And this increase in cytosolic calcium then is going to activate nearby ryanodine receptors, which is going to cause release of even more calcium from the sarcoplasmic reticulum, which then causes another round of activation and releasing even more calcium. So as you can see, this is a positive feedback system, but it's very localized and it's also time limited. So that's the first of three things that are different. The second thing is we have this calcium-induced calcium release. Okay, so backing up the big picture, when the calcium enters the cardiac muscle cell through the calcium channels, these are a type of calcium channels known as L-type, L standing for long because the gate is open a long time. They're also known as slow calcium channels, meaning they're slow to close. So you've got your choice of terminology going on here. So the fact that they're open a long time and slow to close means lots of calcium is going to enter the cell. And so this is why ryanodine receptors are going to be able to sense this rise in cytosolic calcium, because the significant amount of calcium is going to enter the cell. So then we have calcium enter the cell from outside. We have calcium entering the cytosol from within the sarcoplasmic reticulum. And so these two things together are causing what we call this calcium spark. And it's this calcium spark that is what is going to trigger calcium binding onto the troponin for muscle contraction. We'll get to that shortly. But first, the third thing that is slightly different, and that is something called the Circa pump. And Circa stands for either sarcoendoplasmic reticulum or smooth endoplasmic reticulum calcium pump. And so what this is this is an ATPase regulated thing where ATP is going to get broken down into ADP. In other words, energy is going to be used. And the whole point of this pump is to take those high levels of calcium in the cytosol and send it back into the sarcoplasmic reticulum. So in other words, this is doing exactly the opposite of what the ryanodine receptors are doing. All right, so now let's put all three parts together for cardiomyocyte contraction and relaxation. So at the start, up there at number 1, the action potential travels down the sarcolemma, triggering sodium and potassium voltage channels, and then it hits the calcium voltage channel. This is a slow channel. Lots of calcium is going to enter the cell. Cytosolic calcium levels are going to go sky high. Number 3, the ryanodine receptors are going to recognize it. That is going to allow the sequestered calcium to exit the sarcoplasmic reticulum. We're going to have positive feedback locally. We're going to end up with that calcium spark. This then allows the calcium ions in the cytoplasm then to bind to the troponin, to pull the troponin off the tropomyosin, allowing the myosin heads to form the cross bridge with the actin binding site, allowing the little twitch of a contraction. So I've left the words and the numberings on the slide so you can follow the process as we go along. So take a chance to review it now if you don't quite understand it. After contraction, before I move on, as I alluded to before, we don't always need voltage for this to happen. And so there are certain hormones that involve other second messenger systems, for instance, IP3 mediated, and you don't have to know IP3. But these other mechanisms will not involve the ryanodine receptor in order to have this muscle cell to contract. So for cardiomyocyte relaxation, then calcium is going to unbind from the troponin. Now it's back into the cytosol. Those high cytosol levels are going to go over to the Circa pump. Calcium is going to be pumped back into the sarcoplasmic reticulum utilizing ATP, and it's going to be stored there. Now, even after this, we still have elevated levels of calcium because remember, we had lots of calcium come in through those calcium channels. And so the extra calcium is going to go back to the plasma membrane where it is going to be exchanged with sodium. And so for every calcium with its +2 charge we have exit, we're going to have 3 sodiums with their +1 charges enter. Now the problem is sodium's in the wrong place. However, don't forget we still have our sodium potassium pump, which then is going to kick out 3 sodiums and bring back in 2 potassiums. And so you can see the net result of both the calcium sodium exchange as well as the sodium potassium pump is that the charges within the cell are going to stay the same. So take this chance to look at this final slide of this process and review it. This is when we would have a discuss with a partner time. And with that, I'm going to end this first recording.